Okay, so today we'll study the remaining parts of the lipids, and this is class number 11 of BIO201, Introductory to Biochemistry and Biotechnology course. So this is Dr. Didarul Alam from North South University. Okay, so we'll study the remaining part of the lipids, storage lipids. So we studied triacylglycerols are the form of stored lipids. So this is basically a structure. This is basically a stir of fatty acid and glycerol. So to define, triacylglycerol or simplest lipid constructed from three fatty acid molecules esterified with 3 hydroxyl group of a glycerol. So what I said, triacylglycerol, a simplest lipids constructed from three fatty acid molecules esterified with 3 hydroxyl group of glycerol. So there are two types. One is simple, contain one type of fatty acid, and another is mixed triacylglycerol having 2 or 3 types of fatty acid. Look at here, this is one. This is same, but these fatty acids are different. Okay, so this is the primarily storage fat. They are present in many foods. So already I have shown this in the classroom, how to draw the structure or how to write down these reactions. So here 3 molecules of water will be removed. So you can see this is the free fatty acid group, carboxylic group of fatty acid, hydroxyl group and hydroxyl group will react with hydrogen of 3 hydroxyl group of a glycerol, and will release 3 molecules of water and making a covalent bond here. So triacylglycerol provides stored energy and insulation. So in most eukaryotic cells, triacylglycerol form a separate phase of microscopic oily droplets in the cytosol, like here, and serving as depots of metabolic fuel. But in vertebrates, specialized cell called adipocyte stores large amount of triacylglycerol as a fat droplet that nearly fill the cell. In plants, triacylglycerol are also stored as lipid, as oil in the seeds, providing energy and biosynthetic precursor during seed germination. And adipocyte and germinating seeds contain lipase enzyme. That enzyme catalyzes the hydrolysis of stored triacylglycerol, releasing the fatty acid and glycerol for export to the site where they are required as a fuel. The fatty acid released from triacylglycerol are oxidized to produce ATP to carry out energy requiring biological processes. So here you can see the comparison between olive oil's burden and beef fat. So this is the saturated fatty acid, and this is the unsaturated fatty acid, and this is the saturated fatty acid. So this is higher carbon, lower carbon. So you can see this is the olive oil is liquid in the room temperature because of the presence of higher amount of unsaturated fatty acid. But here this is semi-solid because the saturated part is higher than unsaturated part. But this is hard solid because the higher molecular, higher fat is because it contains higher saturated fatty acids, so that this is a fat. So high proportion of long chain saturated fatty acid increases its melting points, so add butters like that. And storage lipids, waxes, are serving as an energy source and water repellent. And biological waxes. What are the waxes? Biological waxes are esters of long chain saturated and unsaturated fatty acid with long chain alcohols. Their melting points is 60 to 100 degrees Celsius, are generally higher than those of triacylglycerol. In plankton, the free floating microorganisms at the bottom of the food chain for marine animals, waxes are the cheapest storage form of metabolic fuel. So waxes also serve a diversity of other functions related to their water repellent properties and their firm consistency. Certain skin glands or vertebrates secretes waxes to protect hair and skin and keep it palatable, lubricated, and waterproof. Birds secretes wax from their clean glands to keep their feathers water repellent. So those are the biological significance or importance of waxes. And also the shiny leaves of holly, rhododendrons, poison ivy, and many tropical plants are coated with a thick layer of waxes, which prevents excessive evaporation of water and protects against parasites. So this is the main major component of beeswax. It is called triacontanol palmitate. This is the palmitic acid, and this is the triacontanol. So writing this structure is very easy. Look at here. So this is 28 and this is 14. So CS3, 14 is double 28 and 1 extra, and then COO. So you have to write first CH3, CH2 14, COO, and CH2 29, CH3. This is quite easy. So let's go for a structural membrane, structural lipid, not membrane, sorry. So basically structural lipids are performed or involved in the membrane formation. So biological membrane is made of a double layer of lipids which act as a barrier to the passage of polar molecules and ions. In phospholipid like glycerol phospholipids and some sphingolipids, a polar head group is joined to the hydrophobic moiety by a phosphodiester linkage. Phospholipids are major component of cell membrane, generally consist of two hydrophobic fatty acid tails and a hydrophilic head consisting of a phosphate group and are amphipathic. So what is amphipathy? That means the compound that can contain positively a neutral charge is called amphipathic. And other sphingolipids like phosphate that have a simple sugar or complex oligosaccharide at their polar ends, these are the glycolipids. So this is the storage lipid. Look at here. This is the storage lipid. So here the triacyl glycerol, triacyl, that means 3 fatty acid and bounded with 1 glycerol. And here membrane lipids are 2 types, phospholipid and glycolipids. Phospholipids are 2 types, glycerol phospholipid and sphingolipids. Glycolipids are sphingolipid and galactolipid. So look at here in the glycerol phospholipid, here glycerol has been attached with fatty acid, and number 3 has been replaced by phosphate and alcohol. So here galactolipids, you can see glycerol has been attached with 2 fatty acid here and here, but this is replaced by monosaccharide and sulfate. So this is quite easier if you compare these two, and you can easily write the structure of those. And sphingolipids, phosphosphingolipids and glycosphingolipids both contain a sphingosine and a fatty acid chain, and where the phosphate group and another contain monosaccharide. Quite easy. So this is another types of archaeobacterial other ethyl lipids. This is also found in the lipid membrane. So they contains much more complex structure, where glycerol is attached with diphentenyl and phosphate, and diphentenyl again bound with another glycerol having phosphate and glycerol. So glycerol phospholipids are derivative of phosphoric acid. So this is also called phosphoglyceride. These are membrane lipid in which 2 fatty acids are attached in a star linkage to the first and second carbon of glycerol, and a highly polar charge group is attached through a phosphodiester linkage to the third carbon. So what happened there? Glycerol phospholipids are lipids where a fatty acids are attached in starch linkage to the first and second, here and here. This is the structure of glycerol: 1, 2, 3, CH2OH, CHOH, CH2. So one fatty acid will be attached here, another fatty acid will be attached here, and a highly polar phosphate group will be attached in the number 3 through phosphodiester linkage. So this is how fatty acids attach with glycerol to produce triacylglycerol. So here, this is the structure of glycerol 3-phosphate and derivatives of glycerol. So glycerol is prochiral and it has no aromatic carbon, but attachment of phosphate group at one end converts it into a chiral compound, so which can be correctly named L-glycerol 3-phosphate. So here are some examples of structural lipids. So this is a common structure of glycerol phospholipid that are named as derivatives of the parent compound phosphatidic acid according to their polar alcohol head group. This is saturated acid, fatty acid, unsaturated fatty acid, and polar head group. For example, phosphatidylcholine as phosphatidylethanolamine have choline and ethanolamine at their polar head group. So this is the X, and it will be replaced by those compounds. So if it is replaced by H, it will be called phosphatidic acid. If it is this X is replaced by ethanolamine, it will be called phosphatidylethanolamine. If it is replaced by choline, it will be called phosphatidylcholine, and accordingly. So this is the general structure of sphingolipids. So what is sphingolipid? The fourth large class of membrane lipids also have a polar head group and two non-polar tails, this and this, and contain no glycerol. So these lipids are composed of one molecule of long chain alcohol, sphingosine, and one of its derivatives, and one molecule of long chain fatty acid and a polar head group that is joined by a glycosidic linkage in some cases and by a phosphodiester in others. So if the X is replaced by H, it will be called ceramide. If it's replaced by phosphatidylcholine or phosphocholine, it will be called sphingomyelin. If it is replaced by glucose, it will be called natural glycolipids, glucosyl cerebrose. Glucosyl cerebroside. So here how structural lipids, sphingolipids are at the cell surface, and how glycerol sphingolipids can determine the blood glucose level, can determine the blood grouping. Okay, so glycerol sphingolipids determine blood groups, and the carbohydrate moieties of certain sphingolipids define human blood group. That means that determine the type of blood that individual can safely receive in blood transfusions. So glycerolosphingolipid or glycosphingolipids are lipids that contain carbohydrate residues with sphingosine as the alcohol and free long chain fatty acid. So this is sphingosine having a fatty acid and carbohydrate molecules. So these are called glycosphingolipid. So the changes in monosaccharide or oligosaccharide content can change the blood group level. So here, these are the O antigen, and if it is elongated by GalNAc, then it will be called A antigen, and if it is elongated by galactose, it will be called B antigen. So presence of antigen will determine the blood group. Okay. So let's go for sterols that are. This is the general structure of a sterol. There are structural lipids present in the membrane of most eukaryotic cell, and this is cholesterol, major sterol in animal tissues, and in plant the major sterol called stigmasterol, beta cytosterol. Major sterol in fungus is called ergosterol. So cholesterol is the major sterol in animal tissues, is amphipathic with a polar head group and a non-polar hydrocarbon chain. So amphipathic means containing the same compound that contain polar and non-polar moieties about long as a 16 carbon fatty acid in its extended form, this one. Okay, so cholesterol accumulated in the blood vessel can block the blood flow, resulting in heart attacks. So cholesterol and other lipids, structural lipids in eukaryotic plasma membrane. So this is the component of this is the schematic presentation of plasma membrane having its different components. Like phospholipid, it contains the 75% of cell membrane, and glycolipid contributing 5%, and cholesterol itself 20%. So in addition to their roles in membrane constituents, the sterols serve as a precursor for a variety of products with specific biological activities. Like bile is a derivative of cholesterol, helps with digestion, and it breaks down fat into fatty acid, which can be taken into the body by the digestive tract. Bile contains mostly cholesterol, bile acids, and bilirubin, and breakdown product of RBC. Bile acids are polar derivatives of cholesterol that act as a detergent in intestine, emulsifying dietary fats to make them more readily accessible to digestive lipase. So, what are the other lipids? Lipids are other lipids contain signaling molecules, cofactors, and energy carriers and pigments. So, lipids with passive roles like storage lipid, structural lipid, we have already studied. These lipids are abundant. Some other types of lipid, although present in relatively small quantities, play a crucial role as a cofactors and signals, and thus are included in the classification of lipids like other lipids. So lipids as signal steroid hormones. Steroid hormones carries messages between tissues. So steroid hormone are any of the group of hormones that belong to the class of chemical compounds known as steroids. They are secreted by adrenal cortex, testes, ovaries during pregnancy by placenta. All steroid hormones are derived from cholesterol. So where here the example of two male and female hormone like testosterone and estradiol, these are derivative of cholesterol. So other lipids like pigment, the visual pigment of vertebrate's eye can derive from vitamin A, serve as a precursor for visual pigments. And the lipid of nutritional importance are triacylglycerol, phospholipids, and sterols. Lipids in the diet transport the four fat-soluble vitamins and assist in their absorption in the small intestine. Okay, so energy storage comparison between carbohydrate and lipid. So these are the carbohydrate and this is the lipid. So carbohydrate belongs to short term of energy storage and lipids belong to long term energy storage. They has more effect on osmotic pressure, but lipid has less effect on their osmotic pressure. Carbohydrate more readily digestible, used for aerobic and aerobic respiration, and lipids are less easily digested and can only be used for anaerobic respiration. Carbohydrate stores half as much ATP per gram, but lipid stores twice as much ATP per gram. Carbohydrates are water soluble as monomers, dimers, easy to transport. Lipids are not water soluble, more difficult to transport. So here is another important thing. Lipids as a cofactor, like lipid quinones, can act as oxidation reduction cofactors. So this is a isoprene units is a branch in unsaturated hydrocarbon, and isoprenoids contain two or many thousand of isoprene units. They are mainly isoprenoid compounds in nature. So ubiquinone, also called as coenzyme Q, and plastoquinone are isoprenoid. So ubiquinone found in the mitochondria of mammals, and plastoquinone found in chloroplast. So they function as a lipophilic electron carriers in the oxidation reaction that derive ATP synthesis. So this is the structure of ubiquinone, and this is the structure of plastoquinone. So ubiquinone is a part of protein and lipids in mitochondrial inner membrane that is collectively called electron transport chain that produces ATP. So most of the usable energy obtained from the breakdown of carbohydrate or fats is derived by oxidative phosphorylation, which takes place in the mitochondria. For example, the breakdown of glucose by glycolysis makes pyruvate, that makes acetyl-CoA, and that enters into citric acid cycle that yields 4 molecules of ATPs, 10 molecules of NADH, and 2 molecules of APDH. Electrons from NADH and APDH are then transferred to ubiquinone and finally to molecular oxygen coupled to form additional 32 to 34 ATP molecules by oxidative phosphorylations. We will study later this electron transport chain. So electron transport and oxidative phosphorylation are critical activities for protein complex in inner mitochondrial membrane, which ultimately serve as a major source of cellular energy. So this is the electron transport chain. We have a more simplified message, a more simplified image in the later slide. So this is the oxidative phosphorylation. This is the metabolic pathway in which cell uses enzymes to oxidize nutrient, thereby releasing energy which is used to reform ATPs. In most eukaryotic, this takes place inside the mitochondria. During oxidative phosphorylation, transfer of electrons are accompanied by expulsion of protein to the outer protons, expulsion of protons to the outer side of the inner membrane, followed by re-entry of proton through ATP synthase enzyme. So complex 1, 2, 3, 4 is involved in expulsion of protons from inner side to outer side, and ATP synthase catalyze the flow of proton from outer side to inner side. The ubiquinone or quinone group allows ubiquinone to function as an electron carrier here. While the highly hydrophobic tail of isoprene unit helps to confine ubiquinone to lipid-rich areas of the cell. So here is the very simplified image of electron transport chain. So this is the intermembrane space, and this is the matrix, and this is inner membrane of mitochondria. So this is complex 1, 2, 3, 4, and this is ubiquinone Q, and this is cytochrome C, and this is ATP synthase. So the concentration of proton is higher in inner membrane space than the matrix. So the NADH that is produced from glycolysis or from TCA cycle contains energy, and NADH releases this energy to complex 1, and as a result, proton from inner membrane from matrix will migrate to inner membrane space, and then electron from FADH will be released into complex 2, and the electron in complex 1 and complex 2 will be collected by ubiquinone Q and transferred to complex 3. And then when electron comes into complex 3, then proton will deflux from mitochondrial matrix to inner membrane space. And it will be utilizing energies because protons is migrating from lower concentration area to higher concentration area, so it requires energy, and the energy is provided by electrons. So those electrons will be collected by cytochrome C and will be drawn to complex 4, and as a result the energies will help the proton to deflux from or to migrate from mitochondrial matrix to inner membrane space. And finally these electrons will be accepted by oxygen and producing water. This water contribute 20% of the water of all the body in our body. Okay, this is how 20% of water is synthesized in our body, and then the ATP is generated here is a higher concentration will migrate through the ATP synthase, releasing the energy, and this energy will be deposited in ADPI and PI bond and making ATP. This is the simplified illustration of electron transport chain. So that's all. Thank you very much for watching and listening.